Welcome, everyone, to the CUSP Roadmap launch. We are going to be sharing our brand new CUSP Research Roadmap here, and we have an exciting program for you with all the 5 CUSP coordinators who will be speaking and a special guest from the European Commission. So, without further ado, I would like to here share the link to the brand new CUSP Research Roadmap. We will also be putting a link in the chat for you to access this document. This has been collaborative work that we started more than a year ago, and it's really amazing and incredibly gratifying to be able to share this now with all of you. Before we get started, I would like to just point out that we are recording this webinar. We will make it available on the CUSP website shortly afterwards. And we also would love to engage with you. So please, while you listen to the presentations and you have a question that you would like to pose to any one of the speakers, please use the question and answer function in Zoom, which you can find at the bottom of your screen, and put your question in there. And time permitting, we will try to get to as many of your questions as possible. And with that, I would like to now move to our first speaker, who is the current chair of CUSP and the coordinator of the AURORA project. Welcome, Roel Vermeulen. The floor is yours, Roel.
Thank you so much, Jane, for the introduction, and thank you everyone for joining this webinar. Just before I start to talk about the roadmap, I wanted to introduce the CUSP cluster to you, which is the European Research Cluster to understand the health impacts of micro and nanoplastics. This cluster was formed by 5 research projects that were funded in 2021 to address this issue, and these projects are listed below here. These were coordinated by Raymond Peters, Tanja Zirkovic, Alba Hernandez Bonita, Mark Morrison, and Rudolf Reuter that have been coordinating these projects, and many of those will actually be speaking to you this afternoon. So why did we actually put a CUSP roadmap forward? In the last 4 or 5 years that these projects have been doing their incredible research, we have delivered many actionable results, and I would ask you to go to the CUSP website to actually look at the policy briefs that actually highlight some of those actionable results. We also realized during this process that although we have made a start, and therefore the name CUSP was actually well chosen, with actually increasing the scientific knowledge about micro nanoplastics, we also realize that there are still gaps and future needs when it comes to micro nanoplastic health research in Europe. So the roadmap that we actually did has the purpose to actually for different audiences, may that be researchers, funders, policymakers, or policy influencers, to look at the roadmap to see what are those needs in the future. This roadmap, as Jane pointed out, has not been an overnight effort. This has taken us about a year to come to this point. We took a very thorough process by first inventorizing basically the current state of knowledge on different transversal topics, and we'll introduce those shortly. As the CUSP working groups have been working on those transversal teams for the last 4 years to summarize that evidence, but also to identify the gaps. That has been synthesized into a first document by a small writing team, which was then discussed publicly at the final CUSP event and also shared to the EU DG RTI for inputs. We received many inputs, which we then have collated back into the document with final editing of all the PIs of the project. So with further ado, I'm going to hand it over back to Jane to actually go into the how and what of the CUSP roadmap. Thank you very much.
Thank you so much, Roel, also for perfect timekeeping. And now we hand over to Raymond Peters, who is the coordinator of POLYRISK and who will give us a little bit more details about what to expect in the CUSP roadmap. Over to you, Ray, please.
Yeah, thank you, Jane. And also thanks for the audience to attend this meeting. We are really excited to show what we have accomplished. And I briefly will tell you how we came to this and what we have done. So we came to these transversal teams based on our results and outcomes. So based on the working group results, so we came to these 5 transversal teams that we will explain more in detail in the next presentations. I will not say too much about this, but the next slide, I skipped this one. The next slide shows you what more or less we have done with each transversal team. So the first one is dedicated to analytical methods and test materials. The second one is on hazard assessment and explains what we have found on toxicity of MNPs. Then the third one is on exposure assessment, where we have explored biomarkers and exposure scenarios. And all CUSP partners have worked on risk assessment frameworks that we will present, and they form a nice basis for future research. Last but not least, we also had some ideas about mitigations, and they're also formulated in the roadmap. And that's with this very brief presentation, I'd like to hand it over again to Jane.
Good. You guys are great at keeping time. Thank you so much, Ray. Okay, now we have heard about why we did the roadmap, what we did, how we went about it. And I think now it's really time to dive a little bit deeper into these 5 transversal themes. And we will now hear from each of the 5 different projects that were involved in CUSP, specifically from the project coordinators, with one exemption, I will get to that, about these 5 transversal themes. And so to kick us off, a warm welcome to Tanja Čerković-Veličković, who is the project coordinator of IMPTOX, or was, I should say, because I think the IMPTOX project now also has concluded. Tanja will share with us the research needs and some key findings from CUSP in terms of analytical methods and representative materials. Over to you, Tanja, and a warm welcome. Thank you.
Thank you, Jane. I'm also very excited to talk about our achievements in CUSP regarding the methods and also the materials we have developed and produced within CUSP. Of course, the key challenging we faced in CUSP when it comes to material production was a fact that these materials are quite diverse when it comes to the polymers, the composition of different chemicals and additives. They come also in different shapes and sizes. However, the reliable test materials and harmonized methods are essential for exposure and hazard assessment to secure comparable results across labs studying micro nanoplastics. Therefore, we put a lot of efforts in CUSP to develop and also use and test those materials. And a range of materials have been produced, polyethylene, polypropylene, polyamide, just to name a few of them, irregularly shaped, some of those aged and labeled. We also should stress that these materials are not inert. They can change under lab or environmental conditions, and they are very needed, particularly well-characterized reference materials with well-defined characteristics such as size, shape, polymer type, and surface chemistry in order to enable exposure calibration in chemical analysis of micro nanoplastics, the toxicology, testing and hazard characterization, and risk modeling. Our approach in CUSP when it comes to the material production is described in a joint CUSP publication on the current good practices for the production of test and reference material. And the principle that guided us was the quality by design approach. In this approach, we need to know the user needs. And then when it comes to characterizing material and producing them, depending on the needs, we can apply a set of a toolbox of characterization methods. So the quality by design inspired approach. When it comes to the analytical methods, standardizing and harmonizing them was done through interlaboratory comparisons. And in CUSP, we have conducted 3 interlaboratory comparisons, 2 with a focus on particle sizing for micro and nanoplastics by different methods. And those studies showed good consistencies when it comes to detection and quantification of micro and nanoplastics. A range of commonly used analytical methods has been tested in interlaboratory comparison, and when it comes to the detection, we can say the methods showed good identification, good results, but the quantification remains challenging. And the results of those interlaboratory comparison studies is also published recently, and you can read more in the published articles. When it comes to the future direction for reliable analysis of micro and nanoplastics, we based these directions on the still remaining knowledge gaps and challenges when it comes to micro and nanoplastics, such as the limited availability of reference materials. Even though we have produced well-characterized test and reference materials, they are not widely available yet. So the immediate needs should focus on developing and sharing the reference materials and quality assurance and quality control protocols to establish reporting standards across labs. When it comes to the midterm needs, we need to improve sensitivity and detection limits of our methods to harmonize sample preparation and the data formats, and the long-term needs should focus on speeding up the analysis, develop high throughput detection tools, and also the overall need is, of course, to integrate exposure, hazard, and analytical data into a unified risk assessment framework. And finally, I should conclude with the message that developing harmonized materials and methods is the cornerstone for understanding and managing micro and nano impacts on human health. With this, I will conclude and hand it over to Jane. Thank you.
Thank you so much, Tanya, for this very, very clear presentation. And of course, it is the cornerstone. That's why it is the first transversal theme in the CUSP roadmap, because without reliable analytical methods and tools, there's no possibility to compare measurement results and to do any of the exposure and risk assessment. So, and also for hazard assessment, of course, it's also very important. Great, so that takes us to hazard assessment. And it's a pleasure to welcome Alba Hernandez Bonilla, who is one of the 2 coordinators of Plasticheal. Plasticheal also now has concluded this year, and it was such a pleasure to work with you, Alba, as with everyone else in the CUSP cluster. So handing it over to you now to please tell us what the key findings from CUSP are in terms of hazard assessment and the key research needs. Thank you so much.
Thank you, Jen. Thank you very much for the introduction. I'm really happy to be here to have some time to discuss with you our advancements in CUSP regarding hazard assessment. So the first thing I would like to highlight here in this slide is that there are many factors affecting the relevance and the reliability of the hazard data. And out of these many factors, I have highlighted here 3. As Tanya has already mentioned, having real-life biologically relevant test materials is really very important. These should be harmonized, and they should reflect the environmental micro nanoplastics in the diversity, the shape, and the surface properties. But it is also really key, as I highlighted here, that these materials are in a size range that is meaningful in terms of hazard. That would roughly mean that this material should be below 1 to 2 microns in order to ensure, and I'm linking to the second point, that the exposure conditions of the systems that we expose and we test reflect really human exposure. This not only means that we should know what is the external exposure to be able to make the correlation in our system, but also to ensure that we have an adequate dispersion, that we consider dosimetry, that we consider also effective concentrations, and definitely that we are sure that the particles that we are testing actually reach and interact with the cells. And assessing cellular and tissue uptake is really, at this point, challenging because we depend on having materials labeled. It is also important that we have a study design that are capable of identifying concentration effect relationships. And as a last point, I would highlight that we need experimental models that are relevant in terms of human physiology, but also regulatory relevant in terms of their requirements. Having said this, we have to admit that not all the studies that we have made in CUSP cover all these important factors, because we started the field when all this was, the knowledge that we had was really limited, and we had to advance in many of these areas. Then there are studies in CUSP that do not use real-life materials. Some others, they do not use these 1-2 microns in size at the maximum. Some do not control all the experimental conditions that are to reflect human exposure, and many of the experimental models that we have used are not harmonized or standardized. So this has made it difficult for us to really have a common message and to integrate all the information that we have generated in risk assessment frameworks that we have developed. But at the same time, despite of the heterogeneity of the studies and the challenges due to this, we have really managed together driving progress in all of each of the single points that I have highlighted previously. All CUSP projects have contributed to building a more coherent and reliable hazard evidence base, because we have advanced in the development of the systems that are in vitro system, in vivo system, and also human studies, biomonitoring studies. We have adapted a lot of protocols and methodologies related to these methods to the micro nanoplastic field. We have also tested the particles that we have managed to generate. We have tested with these systems, and we have measured several biomarkers of effect that are validated. And we have also discovered new biomarkers of effect that can be useful in the future. And finally, we have integrated all the information in the risk assessment frameworks that we have also developed in the different projects. So after all the knowledge that we have generated, we can highlight one of the key priorities that is outlined in the roadmap, which is the need of understanding what are the long-term effects of micro nanoplastics. And this knowledge on the long-term effects needs to be worked progressively from the immediate needs to the long-term needs. We need to develop the system and control all the points that I have highlighted previously in the study design. We have then to test for these long-term chronic effects after long-term exposure. And we need to identify what are the biological mechanisms for these micro nanoplastics, because it's not only important to understand what are the effects, but do it in a way that in the future we can assess the effects of these micro nanoplastics in a predictive way. So as is here highlighted in this slide, if we, after long-term exposure, understand what is the adverse long-term outcome, and we can organize the information in a way that all the effects that we are currently observing in CUSP clusters, like immune dysfunction, oxidative stress, genotoxicity, inflammation, omics alterations, changes in signaling pathways, all these effects have been observed in some of the studies in CUSP. If we can organize the information in a way that we understand what is these molecular events that are leading to the adverse outcome, we would be able to have in the future a way of predicting the long-term adverse effect by measuring just the molecular event that is produced very early in time. Only in this way we will be able to test timely all the diversity of different micro nanoplastics that we have in the environment.
Great.
And with that, I would like to conclude. Thank you to all of you for listening.
Thank you so much, Iva. Very, very important, of course, understanding the hazards and also getting the timeframes right, because exposures are not acute. They're chronic typically. Okay, wonderful. So that takes us right on to the next topic. And here's our surprise guest presenting the CUSP findings on exposure and highlighting the future research needs. We welcome Hubert Dierven, who will represent the exposure working group and also POLYRISK, because our friend Ray is so busy. He had to actually leave. He had to hop up to go to another meeting. So welcome, Hubert, and thank you for helping us out today. The floor is yours.
Thank you very much, and very good that so many people are interested in what we have been doing in CUSP. So that's very good. So exposure starts with analytical methods. That is very obvious. So you need analytical methods to detect these microplastics in biological tissues. So all the CUSP projects have done a lot of work to develop analytical methods to detect these microplastics in human blood, water, and air samples, and also sometimes in human organs. But unfortunately, we still don't have analytical methods available to measure specific nanoplastics. The plastics are less than 1 micrometer. That also was mentioned by Tanya in biological tissues. We have also seen that we need quite large volumes of blood because of the particle issue with nanoplastics. The homogeneity of the particles in blood is variable. So you need either a lot of samples to measure these, or you need larger volumes. And also, what also was already mentioned before, harmonization and standardization of analytical methods is really needed to get reliable exposure estimates. What have we learned? We are continuously exposed to particles in air, food, and water. It's a lot of particles in these media. And the micro nanoplastics, including rubber, is only a fraction of the total number of particles in the air samples. So one of the questions we should also ask is, are these micro nanoplastics more hazardous than the other particles that we are exposed to? We have done in the POLYRISK project a number of studies in Norway, Romania, and the Netherlands. And what struck me was that we see a lot of rubber particles, probably coming from tire wear, from cars and other vehicles, in all our air samples. And we also need to realize that it's not only the particles, but also chemicals that are attached to the particles or that can leach out of the particles that we need to consider. For us in Norway, at least, it's good news that we did a study in bottled drinking water, and there we looked at the microplastics, and we found quite low levels of microplastics in these plastic bottles. But what I can conclude about exposure, looking at all the data that CUSP has generated, but also in other labs, I think it's quite clear that we have plastics in our blood and organs, but how much, that's very difficult to assess. I would personally not trust all the data that has been published on this aspect. We also don't know how long these micro nanoplastics remain in the body, and I think that's also something that we need to know in order to understand the potential health hazards. And I also like to stress as a toxicologist that the presence of plastics in the body is not per definition a health problem. You really need a health effect or an adverse effect in order to say we have a health problem. The problem is these reliable analytical methods for micro nanoplastics are challenging. It's a very heterogeneous group of particles. You have different plastic polymers, you have different chemicals, size, shapes, surface coatings. So it's not easy to come up with one method that fits all. I think we need to have sources of exposure to MNPs should be identified, because then the regulators can do something about it. We need, of course, exposure data in order to perform a risk assessment. So if you look at the roadmap, it's clear that the work is not finished after 4.5 years of CUSP. There's still a lot of work to be done. Immediate need is these reporting standards for studies assessing micro nanoplastic exposure. improving these analytical methods by standard operating procedures and inter-lab comparisons for MNP analysis in real-world samples, so samples from people who are exposed to micro nanoplastics. And we also need to find ways to share our data better across different labs and different countries. And yeah, in the roadmap, also the midterm and the long-term needs are visualized. And ultimate goal, of course, is to do really large scale up, doing human biomonitoring studies in order to follow what is happening with the how large the micro nanoplastic exposure really is, and in what is the direction? Is it going up or going down? I really think that's important, and also to study it with potential health hazards. So that was the exposure part, Jane. Back to you.
Thank you so much, Hubert, and thank you also for highlighting that the micro nanoplastics are present in the human body, and that we still don't fully understand what the associated health risks are related to that. And of course, in the absence of scientific certainty, then it becomes a values preference issue, whether you like to have microplastics in your body or not. So that's obviously not a scientific issue. Good. And with that, I would like to hand over to our next speaker, who is Mark Morrison, one of the two coordinators of PlasticsFatE. And Mark will now have the very difficult task, I think, of tying all of this information together and precisely speaking about what actually the health risks are. So I'm sorry, I preempted your talk maybe a little bit there. But
I don't have to say anything now.
I look forward to hearing what you have to say, Mark. Go ahead, the floor is yours.
Thank you, Jane, and thanks to everybody else who's presented already, and to the large number of people that have come to hear about the CUSP roadmap, which is very exciting and very timely. So, as Jane said, I will talk a bit about the risk assessment frameworks that we have developed within CUSP. And as people will hopefully know, if you've visited the CUSP website, all 5 projects had a slightly different focus and complemented each other. So this briefly tells you a bit about the RIFs that each project produced. So AURORA had a focus on early life, IMPTOX on allergies and asthma. Plasticheal produced a radar plot of specific risk factors and looked at the strength of evidence associated with those. In PlasticsFatE, we produced a predictive risk assessment to support safe and sustainable by design decisions. And in POLYRISK, there was a focus on inhalation based on IATAs. Now, we recognise that there's good complementarity between these RIFs, but there would be a future benefit to bringing them closer together to align them and to integrate them. But at the same time, there are a number of knowledge and data gaps that prevent that, which is the function of the roadmap. So there's knowledge gaps associated with hazard identification and characterization, as we've already heard, and the levels and characteristics of human exposures are still relatively unknown in the real world. And in particular, a lot of our work has looked at models of disease, and we see that there are characteristics associated with MNP exposure that are more of a concern to those populations that are vulnerable or have underlying health problems, which is what I will focus on in the remaining few minutes. So with the risk assessment frameworks, as with the other aspects of the roadmap, we developed a number of actions that need to take place over the next 5+ years, immediate, midterm and long-term needs. And I'm really just going to focus on a midterm need, which is understanding human health risks through epidemiological research. Now, why is that important? Well, most of our observations indicate that there are few acute effects, particularly from pristine micro and nanoplastics. However, when we age those, as you would find in the environment, or we contaminate them with either biological materials or chemical materials, then we see greater effects. We also see greater effects in disease models, particularly disease models where we've looked at gastrointestinal and respiratory diseases, which are two of the primary routes for human exposure. Overall, we've got a very complex picture. So responses are dependent on the type of plastic, the combination of plastics, what biological materials are associated with these, what chemical materials are associated with these, and what particular cells we look at responses. And just focusing on the biological aspects, we see that microplastics can be colonized by a number of different bacteria, but again, this depends on whether that is a pristine plastic, whether there's something else there, whether it's aged or not. So one of our key results and questions for future research is to understand what would be the long-term health impacts of this complex mixture that we're exposed to. So we are recommending that we need further research into looking at collections of real-world plastics, their contaminants, and how people are exposed to it. Looking at different populations, which will include people with underlying health problems, so people that have got gastrointestinal disease, respiratory disease, or are immunocompromised in some manner or other. People that are in different occupations, so people that are exposed or potentially exposed to micro nanoplastics, so in plastics manufacturing and to recycling plants. People in different locations because we know that different locations have got higher underlying exposures, and then overall understanding that as we go forward. And that is me. Jane, thank you very much.
Wonderful. Thank you very much, Mark. I have to admit, it all sounds pretty complicated. And the big question now is, we have all these fantastic results, but we also have a lot more questions, as often is the case with research, more to study. But what can we actually do right now with the knowledge we have right now, and accepting that there are knowledge gaps? What can we do to minimize the potential risk? And to help us understand that, I welcome Roel back as the coordinator of AURORA. Roel, mitigation measures were not really explicitly part of the 5 different CUSP projects, but we realized quite quickly that we had to address that. So please, can you share your thoughts and what's in the CUSP roadmap in terms of mitigation?
Yeah, thank you so much, Jane, for the opportunity to talk about mitigation measures. I'm just trying to progress now. Yeah. So although CUSP didn't focus on mitigation measures, the documentation that there is exposure to micro nanoplastics via air, food, and water, and that we see body burdens, and that there are potential hazards and risks associated with micro nanoplastics, means that we really have to think about mitigation measures as well. That's also emphasized by a recent report where some of the CUSP members were involved in the Lancet Countdown on Health and Plastics. And what was documented in that publication is that the full life cycle of plastics, from fossil fuel extraction through manufacture, use, disposal, and degradation, contributes to disease, disability, and premature deaths across all ages. And it was estimated that per year there's about a $1.5 trillion in health-related economic losses when we think around across the whole life cycle of plastics. And so that's where we not necessarily have to wait till we know all the evidence around micro nanoplastics, but we really need to see how we can actually go to mitigation. And that is mitigation. What we need to do there is to identify the most relevant exposure sources so that we actually can develop mitigation measures and strategies for different exposure pathways. May that be respiratory or oral, and may that be via air, water, and food. At the same time, and this was already highlighted, is that we have to think about what are the safe and sustainable by design alternatives for the current plastics, and those alternatives need to be developed into production. we also have to realize that plastic production has increased considerably over the last decades and is projected still to exponentially increase over the next decades. And if we want to curtail the exposures to plastic across the whole life cycle, including micro nanoplastics, we also have to search for strategies to reduce the production and use of plastics. So these are the key research needs that we identified within the CUSP cluster, and I hope those will be picked up in the coming set of research projects around the globe. And with that, I give it back to you, Jane.
Wonderful, Roel. Thank you so much. And again, thank you very much to all the 5 speakers. We have a bit of time now before we head to our 2 final speakers of this webinar. And so I'd like to open the floor for questions now. And there's a couple of questions that have already come in. So thank you so much to the audience for that. One admin question. Yes, of course, we will share the recording of this webinar. It will be made available on the CUSP website. So check back there, please, to see it then. Okay, so I would like to start with Tanya. Tanya, one question we got was, how are the micro nanoplastic materials selected and how representative are they of environmental samples? And I think that's a really good question because we heard that it's actually the more weathered real world samples that seem to be toxicologically more active.
Yeah, thank you, Jane. That is indeed a very important question. I'd like to say that in CUSP, what we could really select finally was a range of materials that we can characterize in a CUSP, in a cluster. However, because CUSP is made of 5 projects, each of the projects made a selection of the materials they're going to use in toxicity and hazard assessment. Therefore, we finally ended up with a collection of few materials that we can characterize as a CUSP. And I believe that the decision we have made, the selection, was based on the polymers most commonly detected in the environment and in human biomonitoring studies. And also, these materials were prepared in forms that mimic environmental reality. As I have already stressed, we have produced irregularly shaped materials. Also, some of those were aged and additive containing. So this approach made sure that test materials indeed reflected real exposure conditions, not just the lab particles. However, especially during the setting up of the methodology, the pristine materials were, of course, included in testing and in setting up pilot experiments. I hope that clarifies.
Yeah, thanks, Tanya. And maybe just a quick comment, or maybe also a quick question. I mean, there's a lot of toxicological studies using polystyrene nanobeads. And as I understand it, actually, the reason is because those were widely available, or they were kind of the first types of nanoplastics widely available because they were being used to calibrate measurement equipment, right? So, I mean, we're very aware that we have to move on from that because that's an unrealistic human exposure. Okay, yeah, great.
If I can just comment on your comment, Jane.
Please.
We did include polystyrene in our testing. I believe all the projects provided research on the commercially available polystyrene. But what is not really available, what can be further developed, is let's say aged polystyrene. That is not tested yet, has not been tested yet.
Yeah, exactly. Because it's not inert, right? So there will be interaction. Okay, wonderful. Thank you, Tanya. Hopping on to Alba. Alba, there's a question specifically to you. Which analytical method do you think is the best at this point in time to assess 1 to 2 micrometer microplastics in biological tissues? Is there like a golden method that you can recommend?
Yeah, it's a very good but a hard question, because at the moment there are many analytical methods available to do that, but they are really complex and they do not cope. One single one do not cope with all the needs. I would say what we have done in the cluster, we have analyzed this by confocal Raman microscopy, for instance. This is one of the methods that we have find useful to identify these particles of 1-2 micrometers inside cells or in tissue of exposed mammals, for instance. No? The same way, this same technique has worked well in identifying these very small particles, even at the size of 500 nanometers in the blood from exposed workers. No? So this is a possibility. Confocal Raman microscopy worked well for this, but we recognize that there is a lot to do still in harmonizing this methodology, making it more high throughput, to really scale this for saying this is the gold standard. And and what could be very useful for that is interlaboratory comparisons. This is a great tool to really identify what is the best methodology for a given need.
Yes, and we made some good experience with that in the cluster. Yeah,
exactly.
Yeah, of course, high throughput. That's always the challenge. How much time does it take to do a confocal Raman sample measurement?
It takes for one single sample. It could be a couple of days.
Wow.
Work. So if you imagine that you have several tissues or several cell types or several biological samples for a single individual, the number of the time that you need for really having the result is huge. So really, we need to progress on that for sure.
Okay. Time is a good keyword, so let's move on. Hubert, I would like to ask you, actually in that same vein, why is it actually so difficult to measure the internal exposure to micro nanoplastics? And I'm assuming it's not just the time that it takes to do these measurements, right?
No, but you have to realize that, like it was mentioned before, we are not talking about particles that you can see with your naked eye or under a microscope. We are talking about nano-sized particles. So less than 1 micrometer means thousands of a millimeter. So it's really small. It's very difficult to analyze these samples. And also, if you see a particle, then you also need to make sure that it is a really plastic. And that is also not easy to do, because the technology has its limitations, and really to get an assessment of the amount of plastic particles in the body smaller than 1 micrometer is really challenging. So that is a problem. The technical limitations of detecting these particles.
So to summarize, small particle, huge challenge, right?
That's a very good summary.
Okay. Thank you, Hubert. Mark, you mentioned a couple of huge challenges, but in terms of human health risk assessment, in your opinion, after 4.5 years of CUSP, what do you think is actually the biggest or most important challenge that we need to address?
I think the most important challenge is using realistic environmental compositions of micro and nanoplastics. So, I mean, so far we have quite rightly dealt with a number of pristine MNPs that have been aged in pretty specific conditions and contaminated with biological and chemical contaminants. But we don't really have a strong handle on what happens in the real world, where people are exposed to many different chemical and particle pollutants, how they interact, how they affect people that are exposed at a low level for a long time and maybe have underlying disease, or how they affect people that are exposed at high levels but very sort of acute peaks over a period of time. And that to my mind, is where we need to go next. We need to be doing these studies which are long-term and looking at different populations and seeing how we can translate what we see in vivo studies and in vitro studies to these long-term population studies.
Thank you, Mark. And I think that's the key point here. At the end of the day, that's what we really want to understand, the human population sort of broad exposure. And with that, I'd like to turn to Roel. And since you are Mr. Exposome, at least to me, there's a question I'd like to pose to you. Is there any current estimation? What is the exposure for humans in terms of particle per time unit to refer to, so that we can use that when we estimate exposures? I know it's not a strict mitigation question, but
yeah, no, thanks for that question, Jane. And I think this is really where the evidence base is still very limited. And it's limited because of all the aspects that Hubert already introduced, and Alba as well, that it's very difficult to quantify these exposures. And so I think we're at the moment that we can document that these exposures occur, but actually what the level of these exposures are and what those are per time unit is still something that we are largely lacking. And that's where I think the CUSP roadmap, rightfully so, basically focus on QA/QC of these methods, but also on the development of methods that can be used. In the end, if we actually want to go to population studies that Mark was referring to, then we really also have to think about how do we actually scale those methods so that we actually can go to larger sample sizes where we actually can do human biomonitoring, perhaps at scale in Europe, to get a better idea of what these exposures are. Also, especially for vulnerable groups when we think about children and adults. It's important to realize that this is not only then looking at the particles, it's also the associated chemicals and the biological or chemical adhered to these particles. So there are major challenges here still to take, and this will require a combination of methods. And that's, I think, one of the focus points towards the future.
Great, thanks, Roel. And I may just add here that you coordinate AURORA. AURORA is the last of the 5 CUSP projects that actually still is ongoing, because it's a 5-year project, and I'm very happy to be part of the AURORA consortium. So thanks for having me there. And if I may spill the beans a little bit, we're actually working on exactly that right now. Looking at a larger cohort, we'll have, I think it's 700-800 samples, mother-child pairs, that we are busy evaluating now in terms of micro nanoplastics in different samples. Okay, last but not least, before we turn over to our guest speaker, which will be Rita Araujo from the European Commission DG Research. Well, just a quick question to you. In terms of the mitigation measures, how should the different stakeholders, like for example, policy officers like Rita, be involved in assessing the effectiveness of mitigation measures? What are your thoughts on that?
Yeah, I think this is a really important aspect. I think just to reflect back on the last 5 years, one of the observations was that to actually source micro nanoplastics, to know what is used, we actually had quite some trouble to actually identify those. And I think we need to have a real good collaboration also with the private sector and plastic producers to really understand what is going on. When we think about mitigation, we have to think about elimination, substitution, engineering controls, or administrative controls. And we actually need partnerships to really be able to do that. And that needs basically across the different sectors. When we then think about the policy implications, that's really where I think the European Commission plays an enormous important role to think about standardization, to actually put in regulation like the single-use plastic regulation that has been proposed forward, but there are many more other things to do. So I think this is really the key that we have to focus on, and we should really think about how to bring these different parties together within these projects, which we haven't done until now to that scale.
Okay, great. Thank you, Roel. And with that, I'd like to extend a very, very warm welcome to Rita. Thank you so much for taking the time to join us at this launch event today. And Rita, we asked you to share a couple thoughts on the relevance of the roadmap for the European Commission. So over to you now, Rita, and thank you again.
Thank you very much, Jane, for the introduction. Good afternoon. It is a great pleasure to be here today at the launch of the CUSP Roadmap, which is a very important outcome of the joint journey of these 5 EU-funded projects that in the last 4 years did impressive work in studying the impacts of micro and nanoplastics on human health. I started in DGRTD in 2021. This was one of the first clusters of projects I was in contact with, and I must say it was always very interesting to follow the activities, the developments, and the dynamics of the cluster. So I'm very happy to see the final results coming out and the impact, the impact that these results create. I'm trying to change my slides. It doesn't seem to be working, so maybe I need to, let me try to click here. Okay, it works now. So a lot was said already about the scientific evidence and uncertainties on potential impacts of exposures to micro and nanoplastics on human health. So I will just focus briefly on the political context. And I want to highlight that the needs identified by this evidence are reflected in a number of initiatives. Some of these initiatives with the regulatory actions that were taken at EU level in the last 10 years, and they try to respond to these concerns on the impacts of plastic pollution on the environment and also on health. Still, recent assessments by the European Environmental Agency on the progress of the implementation of the Zero Pollution Action Plan show that pollution by microplastics is still far from reaching the desired targets, and this means that exposures will be a great matter of concern for the years to come, exposures to these micro and nanoplastics. As you know, our main instrument to support research at European level are the framework programs of research and innovation. You see here in this slide the current program, in the left side, the current program Horizon Europe that runs until 2027. It is the largest framework program to date. It has 95.5 billion of budget. And on the right side of the slide, there is the proposal that is currently under negotiation for the next framework program. And it has a proposed budget of 170 billion euros, which almost doubles the budget of the program. So this is a very powerful instrument we have at the European level to gather evidence and to develop tools and solutions that can tackle the most pressing scientific needs and also societal challenges. If we look at the trend in funding to environment, climate, and health research, you see that this funding has increased significantly over the last 20 years. But of course, this portfolio includes many different areas, and we need to look to the details to see the trends in this particular area of research. So you can see here in the figure on the right side of the slide, the number of projects funded since 2024, and you see that nanomaterials have received an important share of the funding. But CUSP was, in fact, the first major EU collaborative research initiative that is exclusively dedicated to study the impacts of micro and nanoplastics on health. In this slide, I just want to show the enormous amount of relevant outcomes produced during the lifetime of the CUSP cluster. It is really impressive, and the objective of this slide is just to congratulate the projects for the work, for the good work done that was very relevant to advance this area of research. And I cannot finish without mentioning that this area of research continues to be a priority for us. There is a call in the work program of 2025 of Horizon Europe that is fully dedicated to continuing to investigate this topic. As we could grasp from the presentations until now, there are a lot of knowledge gaps that still need to be addressed in this area, and that is why this call has a long list of activities that we propose to be addressed by the proposals that are submitted to this call. This roadmap that is being presented today is a crucial document to inform, I changed the slide, to inform future research policies in the environment and health portfolio. And I really hope the projects that will be funded under the current call can build strongly on the important work that was done by CUSP. And yeah, I would just like to finish by thanking CUSP for investing the time to bring together this information from the 5 projects that are part of the cluster in this roadmap that really amplifies the impact that the cluster has already created in this research area. And that's all from my side. Thank you very much.
Wonderful. Thank you, Rita. Oh, I almost got a bit sentimental when I saw your slide with all the CUSP outputs popping up. Yeah, we did put a lot of work into that. And I think this is a really good opportunity also to thank everyone who's been involved in CUSP. But I'm jumping ahead because I think Roel would like to do that, right? Roel, back to you.
Thanks, Jane, for the introduction, and thank Rita also for the opportunity that has been given to the 5 projects within CUSP to move this field forward. I just wanted to finish off by first, of course, thanking also the people that tuned in today in this webinar to listen to the results of CUSP and the gaps that we have identified that needs to be taken up in the immediate, midterm, and long-term timeframes to actually understand better the impacts of micro nanoplastics on health. I'm just now trying to move this forward. Yep. So thanks everybody for being tuned in. What we actually intend with this roadmap is that this provides a starting point for funders to identify the research needs that really need funding at this moment. We hope that it helps researchers to guide basically towards where are the identified gaps so that we actually write together proposals that actually help the field forwards to really close these gaps that we actually have identified in these immediate, midterm, and long-term times perspective. There's also a dedication and perhaps a promise of CUSP that we have started this trip together at a time in 2021 when very little was known about micro nanoplastics. Still, there's a lot of road to cover from this trip that we have started. And there's a dedication from the CUSP clusters, the people involved, the researchers involved, but also the PIs of the projects to really take ownership of this trip that we actually take together. I think it would be imperative that with the new cluster, but also the old cluster, that we really look how we actually can do coordination, support actions to keep the research together, because there's a lot to be done. And we cannot do that on our own, and we really have to collaborate. And in that collaboration, we also have to transcend only the knowledge institutions, but really work transversal across policy, the private sector and the public sector to really come up to mitigation measures and evaluate the effectiveness of those mitigation measures. I am incredibly indebted to this road trip that we have taken with over 75 institutions in 21 countries. I think we all started on a road trip where we did not know exactly what the destination was. But instead of thanking actually the PIs of the project, I really have to thank Sabine Kinzel, Catherine Crawford-Brown, Virginia Prodi, Justin Boucher, and Joel Schweitzer that actually have helped in putting this roadmap together, but in particularly also Jane Monkey, who has been facilitating this meeting, but has been key in actually communicating many of the results of CUSP. And with that, I would like to give it back to you, Jane, to close off this wonderful webinar.
Okay, thank you, Roel. And now you made me blush. A huge thank you to all our speakers, especially to Rita and also to Hubert for jumping in. It's always a pleasure to work with you guys. We are not done yet with CUSP. AURORA, as I said, is still ongoing, and we are right now planning a final CUSP event in person, which will be sometime in conjunction with the CTAC Europe conference that's happening in Maastricht in May this year. So it should be easy actually for our colleagues from Brussels to get there. So Rita, please plan to see us there in person and bring your colleagues along. And hopefully many of the CUSP project partners will be able to be there. That's why we're doing it in conjunction with this event that's happening anyway. Please follow us on LinkedIn, check our website, also for this recording. And I look forward to seeing you hopefully in May. And if not there, then somewhere else soon. Take care, everyone, and thanks a lot.